Cell. Cell is the basic structural and functional unit of all living organism. The cell has been variously defined as the unit of structure and function in animals and plants. The smallest living unit capable of independent existence is a small mass of living matter containing a nucleus or nuclear material. History of cell. The cell was first discovered and named by Robert Hooke, 1665, and first saw living cell by Leeuwenhoek's cell theory proposed by Schwann, 1839. Electron microscopic structure of cell. All the cells are made up of protoplasm, a viscid granular substance which consists of water, electrolytes, proteins, lipids and carbohydrates. The protoplasm remain differentiated into an outer cytoplasm and the denser inner nucleus. Surrounding the cytoplasm there is covering called cell membrane. All the living organisms can be categorized into 1. Unicellular organisms, e.g., protozoa, yeast, and bacteria. II. Multicellular organisms, e.g., plants and humans. 3. A human cell consists 100 trillion of cells, size of the cell is 10 micrometers, mass of the cell is 1 nanogram. Cell are two types. Prokaryotic cells. These cells do not have well-developed nucleus, but organelles are present. E.g., archaebacteria. Eukaryotic cells. These cells have well-developed nucleus and other organelles. E.g., plants, animals. Components of cell. A cell comprises of the following three basic components: cell membrane, or plasma membrane, other organelles, cytoplasm, nucleus, Golgi bodies, mitochondria, lysosomes, endoplasmic reticulum, ribosomes etc. Cross-section of an animal cell. 1. Cell membrane, or plasma membrane. The cell membrane is a selectively permeable biological membrane which allows only certain substance to pass through it. Cell membrane is thin elastic and has highly complex structure composed of proteins and lipids. It is a semi-permeable membrane, containing pores, that allow the passage of water, oxygen, CO2 and some solutes in and out of the cell and plays a vital role in maintaining the homeostatic balance of the cell. The cytoplasm consists of a number of structures called the organelles. Right-pointing arrow, it acts as a barrier between the inner and outer surface of the cell. Right-pointing arrow, it also known as plasmalemma. Properties of cell membrane. It guards the cell against the external environment. It allows cellular transport between cells. It helps in recognizing a cell. It divides the cell into smaller sections having different structure and function. It allows the fusion of cell membrane with other membrane of cell organelles through special point of attachment. It regulates the transfer of certain specific molecules through the cell membrane, e.g., gap junction. It enables motility of the cellular organelles. Structure. Fluid mosaic model. The fluid mosaic model is the most accepted one. The fluid mosaic model of the plasma membrane was given by S.J. Singer and G.L. Nicholson. Function of cell. Protection of cells. The receptors present on the cell membrane all chemical messages to pass between the cell and the system, and thus provides protection to cells. Separation from surrounding. In an animal cell, it separates the internal environment from that of the external. Tissue formation. Groups of similar cells together form tissues. Contribution to enzyme acting. It controls the activity of enzyme which further participate in metabolism and immune system. Particle transportation. This occurs by various mechanisms like, carries proteins and ion pumps. 2. Cytoplasm. The cell consists of cytoplasm enclosed within the plasma membrane. The cytoplasm includes various organelles present inside a cell and therefore these organelles are also referred as as cytoplasmic organelles. Basically the cytoplasm is the fluid in which cell organelles remain suspended, mitochondria, Golgi body, nucleus, etc. It is the region lying between the cell membrane and nucleus. The cytoplasm contains cell organ-like, endoplasmic reticulum, Golgi bodies, mitochondria. Structure of cytoplasm. The cytoplasm is divided into three parts, namely cytosol. It is the fluid present inside the cell. The fluid bathes the cytoplasmic organelles. Characteristic features of cytosol include: majority of the cell metabolism takes place within the cytosol. They also act as ribosomes and intracellular receptors. Cytoplasmic organelles. Cell consists the organelles, via Golgi body, mitochondria etc., have some specific characteristics that help in the maintenance of life. Cytoplasmic inclusion. Certain bodies are present within a cell but are unable to perform their own metabolic process. These structures also lack membrane binding them. Function of cytoplasm. It provides the site for cell growth and expansion. It provides space for organelles to remain suspended in the medium. It also helps in the movement of several elements within the cell. Various biochemical reactions, importance for the maintenance of life, occur in the cytoplasm. It has enzymes which help in the breaking the large macromolecule into smaller ones so that other organelles can use them easily. 3. Nucleus. Nucleus is the spherical organelle that consists of a double-layer membrane buildup of phospholipids and protein. It also known as the control cell. It is a vital center of the cell. It controls both chemical reactions and reproduction of cell. It consists genetic material of cell. Structure of nucleus. The nucleus structure is comprised of following compound. Nuclear envelope. A large number of small circular or octagonal nuclear pores are present of specific point on the nuclear envelope. This nuclear pores regulated movement of the macromolecules from the nucleus to the cytoplasm and vice versa. Nucleoplasm. The nuclear cavity of the nucleus is filled up by one of the spiralized forms of protoplasm, known as nucleoplasm. Nucleolus. The nucleus of the cell has a nucleolus also known as nuclei. It is a non-membrane-bound structure containing protein and nucleic acids. Chromosome. The word chromosome has been derived from the Greek word chroma means color, soma means body. A chromosome can be defined as DNA-bound protein, histone protein, that carry genetic information from generation to other. Chromatin material. The chromatic material saturated with DNA transmits the coded structural and functional information between the cell and organism. Interphase chromatin. During the interphase, the period of the cell cycle where the cell is not dividing, two types of chromatin can be formed. 1. Euchromatin. It includes the active DNA. 2. Heterochromatin. It mostly includes the inactive DNA. Function of nucleus. It consists most of the physiological activities of the cell. It functions to encode all the genetic information that carries the hereditary characteristics. It has direct involvement in reproduction, cell division. 4. Mitochondria. Most of the eukaryotic cells contain a membrane-bound cell organelles called mitochondria. Diameter ranging from 0.5 micrometers 10 micro-m. Mitochondria are also known as the powerhouse of the cell, because they generate adenosine triphosphate, ATP, used as a source of chemical. Mitochondria are the chief source of energy in the cell. It is largest cytoplasmic organelles and energy house of the cell as it consists of enzymes which convert chemical energy of the food nutrients into usable energy from TP, which is internally available for cellular activity. It is also known as powerhouse of the cell. Mitochondria is discovered by Albert von Kolliker. It appears like filamentous structure or rod-shaped. It consists of oxidative enzymes which convert chemical energy of the nutrients into form of ATP, and this energy is available for cellular activity. The mitochondria supply 95% of cell energy and so-called powerhouse of the cell. Structure of mitochondria. Mitochondria are double membranous cell organelle having the following parts. 1. Outer membrane. This membrane contains transport protein, which are helpful in transporting ions and atoms across the organelle. 2. Inner membrane. This membrane is involved in the formation of cristae. Cristae are finger-like projection in the matrix bearing small particle called Phi particle or oxysomes. Enzymes of the respiratory chain are present in cristae. 3. Mitochondrial matrix. Matrix of the mitochondria is formed by fluid present within it. This fluid contains enzymes involved in Krebs cycle to oxidize fatty acids and pyruvic acid. Function of mitochondria. Regulates plasma membrane potential and cellular anabolism and catabolism. Synthesis of steroids. Aerobic respiration to create energy for the functioning of cell. The main function of mitochondria, which are organelles also known as the powerhouse of the cell because it is produce energy. 5. Golgi bodies. The Golgi body comprises of folded and compressed membranous sacs piled up each other. All the cell contain Golgi body, however it is large in those cells facilitate the synthesis and export of protein via endoplasmic reticulum to the Golgi complex. They are the double membranous vacuolar channels which traverse the cytoplasm. The membrane appears to be formed of phospholipids, proteins, and a number of enzymes. Golgi bodies act as a sort of intercellular pump that regulates the movement of fluids in the cell and expulsion of secretory products from the cell. Golgi bodies synthesis polysaccharide part of glycoprotein secretion. Structure of Golgi body. The Golgi body consists of membrane-bounded stacks called cisternae. Each cisterna consists of flattened membrane discs that carry Golgi enzymes. Function of Golgi bodies. Golgi body is involved in the synthesis of a different number of macromolecule. Modifying, sorting, and packing. The Golgi complex plays an important role in sorting, modifying and packing of substance for use within the cell secretion. Modification of protein. Proteins delivered by the endoplasmic reticulum are modified by the Golgi body. Transportation of substance. Enzymes present in the cisternae bond to the carbohydrates and phosphate group by the process of glycosylation and phosphorylation in order to modify substance by the transport of substance. Formation of spindle. The Golgi body is involved in the formulation not meiotic and mitotic spindles. Excretion of water. These organelles help in the excretion excess amount of water. 6. Lysosomes. In 1949 a Belgian cytologist Christian de Duve had discovered the cell organelle, lysosomes. The word lysosome derived from Greek word lysis means to separate, educate. They are generally known as suicide sacs or suicide bags. They are minute structures containing enzymes that digest or remove the particles which are useless or may be harmful to the cell. Lysosomes produce hydrolyzing enzymes such as phosphatase acid ribonuclease. Structure of lysosome. Some non-green plant cells possess lysosomes. They are also called suicide bags as they cause intracellular digestion, destruct the carbohydrates, protein, DNA, RNA etc., and thus destroy the cell. Characteristic feature of lysosome. They are present in high number in a single cell. They possess nearly 40 different enzymes. They are referred as suicidal bags since the enzymes present in them can digest the whole cell in case of damage or death of the cell. Function of lysosome. Provide defense against invading microorganisms via circulating macrophages. Helps dissolve blood clots. Cause the sperm to penetrate the ovum. Decompose the mature RBC and dead cells. Help in reabsorption of water and small molecule in kidney and urinary bladder. 7. Microsomes, endoplasmic reticulum. Microsome are a heterogeneous set of vesicles having diameter 20 to 200 nanometers consisting of endoplasmic reticulum. These vesicles are isolated by differential centrifugation and formed three structural features, i.e., rough vesicles, smooth vesicles, ribosomes. These are scattered in cytoplasm. They are two types of endoplasmic reticulum depending upon the presence or absence of ribosomes which are responsible for protein synthesis. Endoplasmic reticulum is two type. Smooth endoplasmic reticulum. Smooth endoplasmic reticulum without the presence of ribosomes. They found lipid. Rough endoplasmic reticulum. Rough endoplasmic reticulum with the presence of ribosomes. They found protein. Structure of microsomes, endoplasmic reticulum, rough endoplasmic reticulum, RER. Small particles of RNA known as ribosomes are found in close association with the membrane of the endoplasmic reticulum, which gives a rough appearance to the membrane. Smooth endoplasmic reticulum, SER. The membrane of SER do not contain ribosomes and help in the metabolism of carbohydrates and lipid. They are involved in the synthesis of steroid hormones in adrenals and testes. Sarcoplasmic reticulum, SR. Most of the muscles consists of a unique type of smooth called sacroplasmic reticulum. Function of microsome, endoplasmic reticulum. It acts as a structural framework of the cytoplasm. It exchanges material with the cytosol by active transport, osmosis, and diffusion. Protein synthesized by its surface-bound ribosomes are collected and stored by RER. It has different metabolic enzymes which helps in the synthesis of phospholipid, cholesterol. 8. Ribosomes. Ribosomes are cell organelles where protein synthesis takes place. They are abundant in living cells. They are either found as free particles, in prokaryotic and eukaryotic cells, or particle attached to membrane as in eukaryotic cells. Structure of ribosomes. Ribosomes are ribonucleoproteins i.e. made from RNA complex and protein. LT is divided into smaller parts called subunits, which are made up of both protein and ribonucleic acid components, smaller subunit and larger subunit. Function of ribosomes. The primary function of ribosomes is protein synthesis. The information carried in the genetic code is converted into protein molecule within the ribosomes. 9. Centrosome. This lies close to the nucleus and is made up of two centrioles, small structures that play a major role in initiating cell division. It is enveloped by a porous nuclear membrane that separates it from the surrounding cytoplasm. During cell divisions, it breaks up and soon after the mitosis it is reconstituted. 10. Chromosomes. They are minute threads-like structure within the nucleus and appear as massed darkly staining material called chromatin. Chromosomes determine the specific characteristics of the cell and hereditary characters pass from one generation to the next generation.